So this is our fourth video on the adaptive immune system. So here we'll talk about cellular immunity. And remember, this is opposed to humoral immunity. So cellular or cell-mediated immunity is a form of specific defense where the T lymphocytes directly attack and destroy diseased or foreign cells. So we already talked about what T lymphocytes, basically what they do, and what the B lymphocytes do. So during this adaptive immune response, the immune system is able to remember antigens and prevent them from causing future disease. There are four different classes of the T cells. These are cytotoxic, helper, regulatory, and memory. So cytotoxic T cells are called killer T cells. You don't need to know exactly what they are with the T8, CD8, or CD8 plus, but do know that they're cytotoxic T cells. These are called the effectors of cellular immunity because they carry out the attack on enemy cells. So it makes sense that they would be called cytotoxic because they're toxic to enemy cells. Helper T cells help to promote the cytotoxic T cell as well as B cell action and innate immunity action. Regulatory T cells, or Tregs if you're cool, inhibit the multiplication and cytokine secretion by other T cells, and they limit the immune response. So they kind of keep it in check so that it doesn't get crazy and cause too much of an inflammatory or immune response. So just like T cells, they can be called T4, CD4, CD4 plus, but you don't need to know these designations. Memory T cells descend from cytotoxic T cells and are responsible for the memory and cellular immunity. Remember, that's unique to the adaptive immune system where they can remember that antigen because they've formed antibodies for them. So both cellular and humoral immunity occur in three different stages. This is the recognition, attack, and memory. So these are the three R's of immunity: recognize, react, and remember. So with recognition, there are the aspects of recognition in cellular immunity are antigen presentation and T cell activation. We already talked a bit about the antigen presenting cells. So antigen presentation is what occurs when the antigen presenting cell encounters and processes an antigen, then migrates to the nearest lymph node, displays it to the T cells. When the T cells encounter this displayed antigen on that MHC or major histocompatibility complex protein, they then initiate the immune response. T cells are able to respond to two different classes of major histocompatibility proteins, MHC1 and MHC2. MHC1 proteins are constantly produced by all cells in the body that are nucleated, which means that they wouldn't be present on erythrocytes, remember, because they're anucleate cells. And they're transported to and inserted on that plasma membrane. So they basically label that this is a normal cell. If they're self-antigens, then they wouldn't elicit that T cell response, remember, because of the positive selection of T cells. But if they are viral proteins or abnormal cancer antigens, they do elicit the T cell response. So infected or malignant cancerous cells are then sometimes, hopefully, destroyed before they can do further harm to the body. MHC2 proteins are human leukocyte antigens, or HLAs, and they only occur on antigen-presenting cells and display only foreign antigens. So again, MHC1 can display antigens that are self-antigens, and T cells can ignore them, but MHC2 proteins generally only will display foreign antigens. So cytotoxic T cells respond only to the MHC1 proteins, and T helper cells can only respond to MHC2 proteins that display only the foreign antigens. So T cell activation begins when cytotoxic T cells or helper T cells bind to a major histocompatibility protein that's displaying the epitope that it's already digested and added to that the major histocompatibility protein. So they will bind to the epitope that the T cell's already been programmed to recognize because it has antibodies against that antigen's epitope. The T cell then needs to bind to another antigen-presenting cell protein that's related to the interleukins so that it can further their response. So this means that the T cell needs to check twice to see that it's bound to a foreign antigen. So that means that it's co-stimulated. So this helps to ensure that the immune system doesn't launch an attack in the absence of an enemy, because that would mean that your own body would be digested or injured, and that's not really good. So that's usually what happens during autoimmune defense. So definitely remember that there are multiple checkpoints to ensure that this doesn't happen. So when there's successful co-stimulation, there will be clonal selection, which means that the activated T cell will undergo repeated mitosis so that it's making clones of itself. And this gives clones of identical T cells that have been programmed against that same epitope of the antigen. Some of the cells after that clone become effector cells and actually carry out the attack. Other cells become memory T cells. So the ones that are effector cells and carry out the attack, remember those are cytotoxic T cells, with memory T cells also being created so that you can remember that antigen. So here is a diagram of T cell activation with antigen receptor recognition, that co-stimulation process, the clonal selection, which means it's just making clones, and then the actual attack. So you have your effector T cells there, and T helper cells that can help stimulate interleukins or stimulate multiple forms of that attack. So during that attack, the helper and cytotoxic T cells play different roles. So helper T cells will play a central role in coordinating cellular and humoral immunity. So they can kind of bridge the gap between that. When a helper T cell recognizes the antigen MHC protein complex, it will start to secrete interleukins that will exert different effects. So they'll attract neutrophils and natural killer cells. They'll also attract macrophages to stimulate their phagocytic activity and inhibit them from leaving the area because you don't want them to go away before their job's done. And then they'll also stimulate T and B cell mitosis and maturation so that you're ready for a larger immune defense. So the role of the helper T cell is threefold, that they can work with the nonspecific innate defense, as well as humoral and cellular immunity. Cytotoxic T cells are the only T cells that directly attack other cells, and this is why they're called effector cells. When a cytotoxic T cell recognizes the complex of antigen and the major histocompatibility complex protein on a diseased or foreign cells, it docks on that cell. So after it docks there, it delivers a lethal hit of chemicals, and these include perforin and granzymes, which kill cells in the same manner as natural killer cells. If you remember, perforin functions to perforate cell membranes of the cells, and so that they can release enzymes that degrade the cells that are called granzymes. Interferons also are released, and these inhibit viral replication. They also recruit and activate macrophages so that they can phagocytize the host cell. Tumor necrosis factor is also very important and is released, and this aids in macrophage activation and helps kill cancer cells, which is why it's called tumor necrosis factor. So after the cytotoxic T cell releases those chemicals, it goes off to try to find another enemy cell while it leaves the chemicals that it left there to do their work in degrading and ruining the host cells. So here you can see what's going on with the cytotoxic T cell and trying to kill off that cancer cell. So immune memory follows primary response and cellular immunity, meaning memory occurs after you've been exposed to an antigen for the first time. So after clonal selection, which is when it multiplies, some cytotoxic T cells and some helper T cells become memory T cells. And these are very, very long-lived. These are up to decades. And they're more numerous than naive T cells, which haven't been exposed to an antigen before. So this means that there are fewer steps for them to be activated so they can respond more rapidly. And this is, again, what's going on when you get a vaccination or if you're exposed to the same pathogen again. So when the T cell is exposed to the same pathogen again later in life, the memory cells launch a quick attack, so no noticeable illness occurs generally. And the person is immune to the disease. Of course, this isn't 100%, and sometimes you do get sick, but it makes it a lot better for you than having the full out illness. So in our next slide, we'll talk about humoral immunity.